Notice that there are numerous proteins scattered around throughout here. Many proteins span the membrane here. It would be called an integral protein, right, or a membrane-spanning protein. And sometimes, although it's not obvious on here, sometimes there are proteins that are just attached to other proteins or whatever like this on the surface. They're not integrated. This would be called a peripheral protein. So an integral or integrated protein here spans the membrane. This one would be peripheral. And they need to be on the outside. This one could have one just sitting right here on top like that. There. On the inside, on the outside, spanning all the way through the membrane there. You see here is some protein here that can serve as an anchor for the cytoskeleton or something like that that's down in there also. So proteins are scattered around throughout the membrane. There's no regular pattern to them. That's why the cell membrane model is called fluid mosaic. It's fluid, meaning that it's flexible, right? And these phospholipids can move around in here. They're not attached. Okay? And it's a mosaic because the proteins are scattered not in a pattern around the membrane. The inside of the membrane looks different than the outside of the membrane. This patch right here looks different than that patch right here. And there are some places where you'll notice sugar that is attached to the cell membrane. In this case here, the sugar is attached to a protein, so this is called a glycoprotein, right? Glyco for sugar is a protein. Here the sugar is attached to a lipid, so that would be called a glycolipid. And you'll notice that if there is carbohydrate, it's only on the extracellular side of the membrane. We never have any carbohydrate on the inside of the cell. And that's because of the function of carbohydrate is one about being a marker or a receptor or something like that. So they mark a cell as a certain type of cell. So they serve as advertisement to the rest of the body what type of cell it is. For example, you know, some people have type A blood, some people have type B blood. The difference between A and B is a type of sugar that's on the surface of red blood cells, a glycoprotein on the surface of red blood cells. Type A have one type, type B have a different type. Type O have neither. Type AB have both. So that's kind of our structure of the membrane. And what the membrane is going to serve to do because of this, it's going to serve to regulate transport. Now, it must be what we call, I'm going to use this word here, selectively permeable. Because if it was impermeable, nothing could come in or out of the cell. So that won't work, right? If it were freely permeable, everything would move in and out, and the internal environment would be exactly the same as the external environment. So that won't work either. So it's got to be selectively. And what makes it selective is the structure and the chemistry of this. For something to move through, first of all, it has to, for the phospholipid bilayer, it has to be small. Right? So anything small can pass through there. Not anything small, but it has to be. Anything big can't pass through. If it's too big, it can't fit through here. Okay, it would disrupt the membrane. It also has to be neutral, because anything that's charged is going to be repelled by this hydrophobic nature, right? This lipids are neutral fats, right? So because of that, things that are going to pass through this hydrophobic layer must also be neutral. So small things, neutral things can pass through. That means that they have to be nonpolar to be neutral, and that means they're hydrophobic. There is an exception, and that's water. Water can pass through freely, even though it is very polar and hydrophilic. Okay? But it is very small. It's got to do with bond angles and things like that, and two partial positive charges and partial negatives. There's a reason why it can move through, but it's the exception. So that's what can move through. What can't move through? So yes, these can. No, these can't move through. Obviously, anything large can't pass through. But even if it's small, like the smallest thing, a proton, can't pass through, right? Proton's a hydrogen ion, right? It couldn't pass through because it has a charge. So ions cannot pass through. Okay. Anything else that is polar, like sugars, amino acids, things like that, they can't pass through at all because they're hydrophilic. Now, you know that even though there are these restrictions, we saw examples where an electron transport chain, right, we saw at the very end, ATP synthase, how hydrogen ions pass through that membrane, right? That's an intermitochondrial membrane, looks just like this, by the way. Okay? But they had to pass through a carrier, right? Remember, it passed through, ATP synthase had a little tunnel through it that these ions passed through, right? So these things, if they're going to be passed across the cell membrane, they have to be facilitated, meaning that we need a carrier to help them move across. Okay, so if we're going to pass something through that is charged, polar, hydrophilic, we need to have a carrier that does it. And these carriers are channels, ion channels. Sometimes we call them transporters. It depends on what's moving it through. Okay. And as a result, this type of movement is called mediated transport or facilitated transport. And there are two types of mediated transport: facilitated diffusion. You may have heard of some of this before, so we're going to talk about these a little bit later. And active transport.